Hello, my dear students. Welcome back to Tamali's classes. In today's class, I've taken up a very important topic of developmental biology of CSI NET Life Science syllabus. And anybody who wants to know, you can also watch this video. So, today's topic is floral meristems and floral development in Arabidopsis thaliana and Anthurium majus. It's a very important topic. So, watch till the end. If you like it, do click on the like icon and do not forget to subscribe to my channel for more such videos of biology. So, let's start. So, these two flowers, as you can see, these are Arabidopsis thaliana. Number one, this one, okay, the first one is Arabidopsis thaliana, and the second one out here, this is your Anthurium. Okay, so I'm written out here raised moss. Don't confuse with it because this flower, Arabidopsis thaliana, it follows a raised moss inflorescence, and the Anthurium plant, it follows a cymos inflorescence. Because if you look at the flowers out here, then you can see, I'll show it to you. So if you look at here, this plant, you can see the flowers. How are they? You can see they are on the lateral sides, right? Okay, and it keeps on growing. It keeps on growing. The growth is indeterminate. Okay, but if you see in the Anthurium, the second diagram, this Anthurium flower, you can see these are all at the terminal end. The flowers are all at the terminal end. Okay, so the growth is determinate. Okay. Now, this is one difference. And if you want to know more about the types of inflorescence, I also have a video made on inflorescence. You can watch that on my channel. Okay. Now, moving on, let's talk about the introduction. Like, let's introduce this topic. Okay. So, what is floral meristem? As I have told you, a meristem, that means it can be either determinate or indeterminate. Determinate means when the growth is comes to an end, when the flowers are growing at the terminal ends. Okay. So what will happen out there? The floral, the growth stage. Okay, when the seed germinates, the plant will, the meristem part, the growing part, will go and rise to the floral organs instead of the leaves. That means, as you have seen in the first one, the Arabidopsis one, see the growth is it's keeping on growing. Okay, it's keeping on growing because um, the floral the flowers are forming on the lateral sides, but here it's talking about when the plant keeps on growing till the end where the flowers are grown. That's called determinate. Okay, because here what's happening, it's giving rise to the flowers and not the leaves. Okay, now transition from vegetative to reproductive phase, that means when the plant is growing till the flower part, and then what's happening due to many other factors like genetic factors and environmental factors, what's happening, the transition is occurring from vegetative to the reproductive phase when the flowers will be formed and again a new generation will be formed. And what are the genes that are that are playing a role in this process? The meristem identity genes, the floral organ identity genes, and also the floral repressors or activators. So what are these things? We'll talk about in our future slides. So let's talk about the Arabidopsis first. So what is Arabidopsis? Okay. So this is a plant. I've told you it's an annual plant. So in this plant, what is happening? See, just listen to me. Okay. Don't focus too much on these slides. I'll tell you when you need to look at the slides, or even like if you want to take up notes, you can take up the notes. Okay. So when a seed is germinating, now this is germinating, the shoot will develop into the leaves, and the leaves will develop into the reproductive part. So that is the process of growth of a plant. Okay. Now, during this, two genetic changes are occurring. Number one, the floral induction from the vegetative to the floral state. Because first, you do not get the flowers immediately. First, you get this, the stem will grow, the leaves will be forming. Okay, so it will generally, it will gradually, it will lead to the floral state. That's the number one step. Number two step is that when the floral state is achieved, the floral development occurs. That means the different parts of the flowers are occurring. That means the plant is committed to form the floral stage, or you can say flowers. Okay, now this floral development comes again, there are two parts. Number one is when the meristem identity changes, when the floral meristem is growing. Okay, that is from identity of the shoot to the flowers. And number two is the floral organ identity change, that means the floral parts. Okay, we know there are many worlds in the flowers, like we have the sepals, we have the uh, petals, we have the stamen carpels, right? So these are all different floral parts. So first is the growth of the shoot to flowers, floral meristem identity genes, and second is the organ identity genes. Okay, now Now look at this slide out here where I have mentioned all the classes and genes. Now, if you look on the left hand side, the floral, meristem, and genes, here there are two classes, okay? Class 1 and class 2 of genes. Now, class 1 has got three genes, leafy, apitella 1, and cauliflower. And for class 2, there is one, that is terminal flower, TFL. Okay, so they will maintain the identity of the shoot meristem. Now, coming to the floral organ identity genes, now there are three classes A, B, and C. You have to remember this very carefully because in our next slides this will be there. Okay, so in class A we have two, Apitella 1 and Apitella 2, and in class B we have got the Apitella 3 and Pistillata, and in class C we have got the Agamous gene. So in these all these genes they have got different roles, and I'll tell you what they are. Okay, now coming to the meristem identity genes. So first we said I told you about the meristem identity genes. So what are the genes that are responsible for the meristem identity? Okay. So if we look at the two flowers, Arabidopsis and the Antirrhinum. Now see, for Arabidopsis there are two genes out there, leafy genes and Apitella 1. Leafy, what does it do? It will promote floral meristem identity. Now if we talk about the Antirrhinum plant meristem identity genes, then we have got again two, Floricola and Squamosa. Okay, Flo and Squam. So these two are just the orthologous to the previous ones. That means the Flo, okay, Flo gene is orthologous to the leafy gene, and the Squamosa or Squa genes, it's just the orthologous to its orthology of the Apitella 1. Okay, ortholog. So remember this. So what does Apitella 1 does? It will specify the floral meristem and also it will help in the identity of sepals. Remember, it has got two functions, floral meristem and also sepal identity of Apitella 1, AP1. Okay. Now, suppose, this is very, very important, it comes in the exam. Suppose there are mutants, that means there's a mutation in these genes. So since LFY and Flo, since they are helping in the floral meristem identity, what will happen? No flowers will be formed. Okay, they will form only leafy shoots if there is a mutation. Okay, if there are mutants. Now AP1 and squamosa, if there is a mutation, what will happen? So this mutation will lead to defective floral meristem. Why? Because they are forming the floral parts. So if there is a mutation, it will not form that. Okay, so they will form defective floral parts. Remember that. Okay. Now coming to the floral organ identity in Arabidopsis, ABC model, very, very important again, ABC model. So as I've told you in my previous slides, I will tell you the function of each ABC classes. Now see here. Class A, okay, Class A, Class B and Class C. Class A will be responsible for the sepals. Okay, remember. Okay, look at here. See. So class, okay, here they are talking about gene ABC. Okay, let's talk about gene ABC. If you see gene A out here, okay, if you talk about class A is responsible only for sepals, okay, and class B is responsible, not class A, gene B, wait, okay, yeah. So class A is responsible only for sepals, B is responsible for both petals and stamens, okay, and carpel is for, gene C is for carpels. Just remember that much, don't get confused, I'll tell you that in details, okay. Now, first of all, we're talking about the organ identity. So I told you about in Arabidopsis there are class A, class B, and class C. And genes are out here. What are the genes? Genes are AP1, AP2, AP3. Okay. Now, just remember one few things. Just remember first, and then I'll tell you that in details. Okay. So this organ identity was discovered by Elliot Meyrowitz and Enrico Coin in 1991. One thing. Number two, as I told, there are three classes A, B, and C. Just before going to the next slide, just remember that A and C are antagonistic in nature. As I told that AC is the sepals part and CC is the carpel part. So when A is mutated, when there is a mutation in A, it will lead to formation of C, and when there is a mutation in C, it will lead to formation of A. Remember that, okay? A and C are antagonistic. So when sepals, there is mutation in sepals, it will form carpels, and when there is a mutation in carpels, it will form sepals. But B is independent of both A and C. Remember that, okay? That means it will not matter if B is there or not there, whatever, because if A and C are antagonistic, but B is independent of both A and C. So how that is, I'll show you. Okay, now remember, A is for sepals, C is for carpels, but B, A, okay, now B is in combination with A and C both. Okay. If you look at the diagram out here, this B, okay, B has got both this petals and stamen. So B with A will form petals, okay, and B with C will form stamens. But for sepals, it's only A. Remember this, okay? See, for sepals, it's only A. Fine. For carpels, it's only C. But when we talk about petals, it's both A and B. Then we talk about stamens, it's both B and C. Now, are you clear about it? See, don't worry. If you talk about on the basis of words, okay, don't talk about on the basis of genes. Remember this, for sepals we have got A only, for petals we have got both A and B, for stamens we have got B and C, and for carpels we have got only C. Okay. Now this is very important. Here I have mentioned about the functions of the petals and about the gene classes, and also what are the genes involved, also how you specify them. So if you look out here, this A, gene class A, have got sepals and petals both. Why? Because if we're looking at the function, see, I told you, if we talk about the voles, okay, if we talk about the words like sepals, petals, stamens and carpels, then for sepals we have only A, but A is there for both sepals and petals. But for sepals only A is, remember. Don't confuse this, okay? I know you will confuse this. So please understand it very carefully. For sepals, it's only A, okay? But for petals, both A and B is there. Fine. For carpels, there is only C. But for stamens, both B and C are there. Therefore, out here it's written like this, that A is present in, like A is having function of identity of both sepals and petals. B is having identity of both petals and stamens. And C is having identity of both stamens and carpels. Okay. Now, what are the genes involved? As we know that class A has got AP1, AP2, class B has got AP3 and PI, and class C has got AG genes. AG has a very important function. I'll tell you that in my next slides. Okay, so remember this organ specified, the how you show it. Sepals only A, as I told you. See, petals A plus B, again petals A plus B, stamens B plus C, but again stamen B plus C, but carpels only C. Okay, don't confuse this. Now, mutants in cluster of Arabidopsis. As I told you, I remember I told you about this one, about how A and C are antagonistic. This you have to remember out here, okay? Remember this, A and C are antagonistic, but B is independent of A and B is independent of C. Fine? Now I'll tell you how. See, this out here, the first one out here, okay, Vol 1. In Vol 1 is for sepals, okay? In normal flower, it will be A sepals. In Vol 2, normally it will be petals, A plus B. Vol 3, normally B plus C stamen. And Vol 4, normally it is C is carpel. What's the phenotype out here? Phenotype means the external expression, okay? The phenotype out here is SE sepals, PE for petals, ST for stamen, and CE for carpel. But suppose it's a A class mutant, that means there is a mutation in the A class. Now what will happen, as I told you, A and C are antagonistic. So out here in place of A, C will be there, carpel will be there. Okay. Now in place of A, I told you C will be there. So C, A plus B will become C plus B. Okay, B plus B will remain B plus C because there is no mutation in B and C, and C will remain C. So that is how the phenotype will change. Okay, so out here the first one SC will become this CA, and even out here this one will become ST, that means stamen. Okay, now so what happened when there was a A class mutation? Sepals turn into carpels and petals turn into stamen. Remember this. Okay, now coming to the B class mutant. Suppose there is a mutation in the B class. What will happen? The A will remain A sepals, but out here A plus B, what will happen? A plus B will remain sepals because there is a mutation in B. But since B is independent, so B will not change into A, B will not change into C. B will not be there. Okay, so only A means sepals. Now again B plus C, B is again there's a mutation in B, so no B, so only C. C means carpel, and the last C will be C. Okay, so what happened out here? B class mutation Sepals became sepals, petals became sepals, stamen became carpels, and carpel is carpel only. So out and no change in sepals and carpels, but in Volt 2 and Volt 3 there is a change. Now, suppose it's a C-class mutant, okay? Sepal C-class means the carpel part. So as we know, A and C is antagonistic. The carpel will change into sepals 1. Also here also we had B plus C in Volt 3, okay? So again C will change into A. So now it will become petals. And now Volt 1 and Volt 2 will remain as it is because there is no mutation in A and B. Now what did happen? In C-class mutants, but the Volt 3 from stamen it became petals, okay? And from carpel it changed to sepals. Even the phenotypic expression changed. See, SE sepals, petals, petals, sepals. Even in the B class, sepals, sepals, carpel, carpel. So this is the mutation table, and you have to remember this for your exam because questions come from this part. Now, coming to the ABCDE model, this is a revised ABC model. Okay, this was given. This is also known as the quartet model. It was given by Thiessen in 2001. Okay, so out here you can see A, B, C, D, E. So what has come? D and E has come. Okay, now D is said to um, play a role in the ovule identity. Okay, and what are the genes responsible for that? We have got shatterproof and seed stick. Okay, again very important question can come from here. Shatterproof is SHP and STK. So these two genes are responsible for ovule identity. Now coming to E. E is that, what is E? See, E is a organ combinatorial entity. That means E will be present with all the other classes in combination with them. Okay, that means A will be A plus E, B will be B plus E, C will be C plus E, D plus E, and E. So that means what? In E we have got C palata genes. Okay, it can be from 1 to 4. This gene number. Okay, so C palata genes, they act as combinatorial partners with A, B, C, and D. For what? To specify the particular organs. Okay, so it's just similar. You can see the diagram out here. Same. Okay, see A. A is showing both sepals and petals. The sepals has only A, okay, but A is showing both sepals and petals as we have seen in the previous. But here what is adding to it? E, because E is in combination with all the classes. So out here A plus E, A plus B plus E. Okay. Now see the stamen part. Stamen, we know that B plus E was there, right? So now what happened? Who added? E came. So B plus C plus E. Okay. Now see here. B class is showing both petals and stamen. Okay. And C class. C is showing both stamen and carpel, but carpel is can be shown only by C. Remember, carpel only C, sepals only A. But A is having to A shows both sepal and petal, and C shows both stamen and carpels. Remember that. Now, since in this ABCD model we have D and E, so these are going added here for ovules. But you can see E. E is there for all of them. Okay, E is there for all of them. So E will be added to all. C plus E, C plus D plus E, all is there. Okay, so this is your ABCDE model. Okay, see C is also showing a part in the ovule development also. You see here, so C plus D plus E. Fine. Remember the genes. Okay, remember the genes. Now in anterorhinum we're talking about the anterorhinum. Arabidopsis is over. Talking about the anterorhinum part. In anterorhinum, same process. Okay, it's similar to Arabidopsis. Here, the same thing happens. Transition from vegetative part to reproductive part. Okay, vegetative means what? Leaf, stem, shoot. This all will go to the reproductive stage. Okay, so that means the shoot apical meristem will turn into the inflorescence meristem. That means flowers. Okay. Also, there is a floral meristem initiation. That means it will give rise to the bilateral symmetrical flowers. Now here there is another what is the difference between the Arabidopsis and Antherium is the genes. Okay, other genes. So here we have again outer flow. Okay, FLO, floricula. Floricula is similar to LFY in Arabidopsis. Okay, that means it is helping in the meristem part. Okay, so again same, it has got A, B and C genes, similar like the Arabidopsis part. Again it has got the ABC model also. Okay, all are same. Now coming to the ABC model out here, and let's talk about the genes. See, in Arabidopsis, what were the genes that we had? See, in Arabidopsis plant we had this AP3 gene, AP3, PI and AG, right? Now, just in anterenum, we have the genes that are similar to the ones of Arabidopsis. We have got deficiency DEF for AP3, okay, and then GLO globosa. We have got in place of PI, and we have got plena PLE in place of AG. Okay, that means okay. One more thing, squamosa. Squamosa I told you in the beginning. Okay, squamosa is a function gene. Fine. In Arabidopsis, what did we have in A function gene? We see here. See, we have out here in A function out here. We had got apitella 1, apitella 2, right? So here we have got squamosa, okay, A function. And then we have for the petal and stamen, we have got deficiency and globosa. So A function, okay. Squamosa have got again two functions. Number one, it has got in the floral meristem identity, and number two, it has got for the sepal development. Remember this, okay? Don't forget. Squamosa have got two functions, sepals and floral meristem, okay? Then we have got deficiency and globosa. Deficiency and globosa are involved for the petal and stamen development. They have the function. These genes are involved in these functions. Then we have got plena. Plena is a C function gene. Okay. So what was C doing out there? Okay. So out here same thing, but here it's plena. Okay. Plena, what does it do? It's involved in the stamen and carpel development. Remember, carpel has got only C, but C has got both stamen and carpel. So plena out here, plena genes takes care of both carpel and stamen development. Okay. Now same thing again, similar to the previous one, A, B, C, D, E model in anterenum. Now, as I told you, A has got squamosa, B has got deficiency and globosa, C has got plena. D function is not so much studied in anterenum, but it is because basically D is for ovule development. Okay, so it is there, but it's not studied in anterenum. Okay, then we have got E genes. E function genes we know only it is in combination with all the other genes. So out here E genes is the lipless gene, remember. Okay, E genes is out here the lipless genes. It will function like similar to the previous one, Arabidopsis, but here this is the in anterenum the lipless genes are considered as to have the E function. Okay, now let's see what are the effects if there is mutation in these genes. This is very important question for your CSIR exam. Now in squamous we know it's helping in the sepal development and also the meristem development. So if there's a mutation, loss of function, okay, what will happen? Sepals will convert into carpels because I told you A and C are antagonistic. Then def and globe, okay, these are both homeotic genes. B plus, remember. So if there is a mutation out here, then either one or both there will be loss of function. So petal will turn into sepals and stamen will turn into carpels. We have seen the in the table, remember in the previous one. Plena is again B and C because it takes care of both the this carpels and the stamen out here. So what will happen if there's a mutation in this plena B plus C? Stamen will convert into petals and carpel will transform into the sepals. Okay, and lipless E. So lipless again, if there's a loss of function in lipless, if there's a mutation in lipless, what will happen? Since lipless is in combination with all the others leading to formation of the floral organs, what will happen? There will be absence of floral organs because there is a mutation in the lipless gene. Okay, so instead of flowers, what will happen? There will be some defect and leaf-like structures will be formed. Okay, so this is the phenotype one, the right hand side. Fine. Now, coming to the table, I have shown you a table. It's just similar to the previous one. Just to make it clear to you, I have done this. Okay. So see, in wall 1 we have got A, sepal squamosa, B we have got deaf and glow, C we have got stamen, and also in C we have got carpel. Okay. So again, if there's a class mutation, what will happen? A, where is the A? A is in the first one, wall 1 will become carpel. Okay. Also, since there is no A out here, the second one out here, see it's written stamen only. Okay. But stamen only is there, but actually A is also involved in this wall. Okay, so what will happen? A will become again carpel, remember that. Okay, so but here since there is not written, since B and C is more prominent in this wall too, so here only stamen has been written. Now number C, again if there is a this see A is the mutation has occurred, not B and C. Okay, so outer no changes. Again here see if there is a B mutation, where is B present? Out here. Okay, since it's responsible for both this one sepals and carpels. What will happen? Because of stamens and semen petals and stamens. So because of B mutation, it will lead to formation of sepals and carpels. Okay, and the rest will be same only. Okay, okay. Again, in this one, see B plus C. Okay, in the B plus C out here, it will form again. Since there's a B mutation, so B will be removed. Only C will be there. That is, only carpel will be there. Okay. Again, last one will remain carpel only. In C class, the last one, carpel will become into sepals, and the second last one, where B plus C was there. Okay. Out here, since C is there, C will change into A. Okay. So, but out here, will form petals. So petals and carpels was there, but here it will form only petals. Okay. In C class mutation. So if you see, sepals became sepals. A sepal is only there for petals and stamens. Only petals are there because there's a C mutation out here. Then this is the same thing. So do one thing. Instead of, this is a bit confusing, I understand that. So you can just refer to the previous one. Okay. This one don't look at that much. What I'll do is I'll delete this slide so that you don't get confused in this one. Okay. So I'll delete this slide so that you don't get confused. Just remember this one. Okay. Now coming to the regulation of floral meristem determinacy. So this is again very important. As I told you previously that there is a very important function of AG genes. So AG out here, AG performs two functions. One is that it specifies stamen and carpels, okay? And it also represents the Wuschel gene. What is Wuschel gene? Wuschel gene is that gene which is responsible for the growing or for the maintaining of the stem cells, okay? If Bush gene is not controlled, if it is not repressed, then the plant will keep on growing. It will be the plant will become will have the indeterminate growth. Okay, so Bush has to be repressed by the agamus gene. Okay, so as I told you, just show you here. I've just mentioned this same thing out here. So agamus function I've given out here, and also what does Woos do? Woos will Woos will help the meristem to keep on dividing, okay, and we will keep on growing extra floral organs and indeterminate growth. So that is repressed, or that is suppressed by the, or you can say inhibited by the agamus gene, okay. Thus it will make the plant determinate, it will make the flowers determinate. That means it will not keep on dividing and growing, and that is required, okay. Now what is the difference between Arabidopsis and Anthurium? I've just mentioned here. Growth habit is, it has got a rosette and this is erect, okay. And then in inflorescence, I've already mentioned in the first slide, Resmi and Cymos. In then B gene, in B gene, Arabidopsis has got AP3 and PI, whereas Anthurium has got DEF deficiency and globosa GLO. In C gene, we have got in C class we have got AG gene in Arabidopsis and Plana gene in Anthurium. For A class, we have got AP1 and AP2 genes in Arabidopsis and SQA and LIP1, LIP2 in this Anthurium. Okay. Similarly for meristem, and see these three. Okay. See these three. Like, what's happening? Okay. See these three are the floral identity genes. Okay. This is the meristem identity gene. Last one. The meristem we have got LFY AP1, and in Anthurium we have got flow and SQA. So let's summarize the entire process. What do we know? We know that first of all, there's a meristem, vegetative meristem. Okay, it's called as shoot apical meristem. This will be formed after seed germination. Then it will there'll be transition from vegetative meristem to the inflorescent meristem. Okay, and then there'll be the initiation. How will it be occurring? With the help of the LFY and AP1 genes. Okay, and then we have got the organ specification. That means ABC genes. When it goes to the inflorescence stage, the ABC genes will play their role. And then what will happen to control that growth, to control that this thing, meristem growth, or you can say floral growth, there has to be some genes, okay, that is called, out here we have got the AG genes. It will replace the WOS genes, and they will be meristem determinacy achieved, okay. This is the summary of the floral developmental stages, and these are some questions which I have given for you so that you can answer these questions and see if you have doubts or not. This is a very important question, very easy and very important questions also. So what are the big floral identity genes? Phenotype of AG mutant. Then how does AG regulate floral determinacy? And also match the following. Okay, match the following. I've just given the question, but not required. But you can do that. You can match it and you can say what are the functions and also which gene represses Bushchild gene determinative. This is very easy. Okay, and thank you for coming till the end of the class. Okay, for watching the video till the end. And if you like the class, do click on the like icon, share it with your friends whoever need it, and subscribe to my channel for more such videos. That will help me a lot and also motivate me to upload some more such videos for your knowledge. Okay, so have a very nice day. Bye bye.